At Family Tree DNA, you can view both the Y-DNA and the mtDNA haplotrees. Let's take a deeper look into what the mtDNA haplotree is, how it works, and how to read it. Everyone on the planet has inherited mitochondrial DNA from their mother and their mother's mother, and so on. By testing that mitochondrial DNA, also known as mtDNA, you can discover more about your direct maternal lineage. The current version of the mtDNA haplotree is the Haplotrea version 17. This was built with a little over 24,000 academic and privately submitted samples, some of which only had partial mtDNA results. It has 5,438 branches and was last updated in 2016. Tree DNA's Million Mito project is picking up where the Haplotrea left off and examining all mtDNA full sequence results to build an updated tree. Our mtDNA full sequence test scans your mitochondrial DNA to detect small differences that distinguish each lineage. By putting them all together, we can create a huge family tree of all maternal lineages on the planet. We call it the Tree of Womankind, or the mtDNA haplotree. While the major updates are still on the horizon, Family Tree DNA has already added more branches and has almost half a from the Family Tree DNA and the Genographic Project databases to analyze and help build the new version of the mtDNA haplotree. It's 20 times more sequences than the Haplotrea. In 2022, the Million Mito project released the first update to the mtDNA haplotree since 2016, a previously unknown 100,000-year-old lineage. You can read all about this exciting discovery on the Family Tree DNA blog. Many more branches will be discovered and released in the future, providing more granularity to your mtDNA results. Let's take a look at how that process works. The process for building the mtDNA haplotree is currently different than how the Y-DNA haplotree is built. The Y-DNA haplotree is defined and built by every new big Y result. New branches are added to the tree and inserted into their proper place as they're discovered. The Haplotrea doesn't work like that. It was comprised of sequences from academically researched samples and periodically updated over the years, with the last update happening way back in 2016. Branching for the mtDNA haplotree is designated by alternating letter and number formats, like how the Y-DNA haplotree was over a decade ago before the SNP explosion from the big Y. Your branch and your haplogroup will look something like L7A1A or H3B6A. Mutations are assigned to each new letter or number. So if you're H3B6A, you have the mutation or variant required for each step down the branch. H, H3, H3B, H3B6, and H3B6A. The Million Mito project was launched in 2020 as a collaborative effort between Family Tree DNA's research and development team and the scientific portion of the Genographic Project. The goal is to pick up from where the Haplotrea left us in 2016, analyze more mtDNA sequences, and reimagine the mtDNA haplotree. By examining more mtDNA data and applying the process that allowed Family Tree DNA to build the world's largest Y-DNA haplotree, the Tree of Mankind, we can also create the world's largest Tree of Womankind. For an in-depth look into this research, there's a great video from Family Tree DNA's population geneticist, Dr. Paul Mayer, from RootsTech 2022. It's still available in the on-demand section in the RootsTech site. If not all of the haplogroups and branches from the Haplotrea are very old. The time to most recent common ancestor, or TMRCA, for the branch is estimated at thousands of years ago. Just matching someone with the same haplogroup as you is not helpful for determining when your genealogical common ancestor was or who they were. The Million Mito project is building the mtDNA haplotree by using new sequence data, 20 times more data than the Haplotrea used. That data will create new branches and, most importantly, younger branches. The more refined branching on the new mtDNA haplotree will be able to provide you with a better TMRCA with your mtDNA matches, with many estimated within a genealogical time frame. We can look at this example of haplogroup U5A2B2A. On the left, the before image is from the Haplotree. With the Haplotree, there's barely any definition downstream of this branch, and the branch is estimated to be 5,000 years old. On the right, the after image is from the Million Mito Project's research. Analysis from all of the additional sequences at U5A2B2A today allows us to refine this branch further into new subclades that have a TMRCA estimated between 2,500 years ago to as close as 500 years ago. Additional sequencing, meaning more testers with MT full sequence results, will allow us to refine these subclades and the TMRCA even further, like we already do today for the Y-DNA haplotree. Now, let's take a look at how to read the mtDNA haplotree. The haplotree can look overwhelming at first, but reading the haplotree is actually pretty easy once you get the basic terminology down. Let's start with haplotree itself. For mtDNA, this is the tree of womankind, the family tree of all direct female lineages. A SNP is a single location or a mutation on the mitochondria. A branch is where a SNP has been placed on the haplotree. These three terms have to do with the SNP's placement on the haplotree in regards to the other SNPs on the haplotree. Upstream SNPs are ancestor SNPs. They're the parent, grandparent, great-grandparent, and so on of another SNP. Downstream SNPs are descendant SNPs. They're the child, grandchild, great-grandchild, and so on of another SNP. Sibling SNPs share the same parent SNP. Upstream denotes the parent or ancestor of a branch. A branch that's upstream of another branch will be placed above them, similar to how your parent or grandparent would be placed above you in the family tree. Let's look at the branch with the middle arrow here. It's H2T152C exclamation point. You can trace the line descending from the bottom left corner and see that it splits to the right into H2C. This indicates that H2C is a child branch of H2T152C exclamation point, and therefore H2C descends from H2T152C exclamation point. So H2T152C exclamation point would be considered upstream of H2C. The same type of connection is not shown between H1, the top arrow. No line descends from the bottom of that node into H2T152C exclamation point. Although H1 is visibly higher on the tree, it's not directly related to H2T152C exclamation point, and therefore would not be considered as an ancestor of or upstream of it. Downstream denotes the child or descendant of a branch. A branch that is downstream of another branch will be placed below them, similar to how children would be placed below you in a family tree. Using the same section of the haplotree, H2C, the bottom arrow, is considered downstream of H2T152C exclamation point, though not downstream of H1. Also, any descendants of H2C, like H2C1 below it, would be considered downstream of H2T152C exclamation point, as they're all connected through H2C. Siblings are two or more branches of SNPs that share the same parent branch. Sibling branches are placed next to each other and underneath their parent branch, similar to how you and your siblings would be placed next to each other and under your parents in a family tree. Let's look at H1A. There's a line that extends down and out to H1A1, H1A2, H1A3, H1A4, H1A5, H1A6, H1A7, H1A8, and H1A9. This indicates that all 9 of those branches are descended from H1A. They're downstream. Those 9 branches don't have lines that vertically connect each other directly. They're only connected by the line that descends from H1A. This indicates that while they all share the same parent branch, they're not upstream or downstream of each other, meaning that they're sibling branches. For more information and additional help with the mtDNA Haplotree or the Million Mito Project, check out Family Tree DNA's Help Center at help.familytreedna.com, and you can also contact our customer support at www.familytreedna.com/contact. I mentioned Dr. Paul Mayer's 2022 presentation on the Million Mito Project's research earlier, and that can be found on the Roots text set.